So our first guest, and now I become a moderator almost, is Professor Jack Heinemann. He's a professor of genetics and molecular biology in the School of Biological Sciences, and he's the director of, what do you say, your lonely center for integrated research in biosafety at the University of Canterbury in New Zealand. So, you come up. Then the next is Professor Ignacio Chapela. He's as you say it, a scientist by conviction and an aspiring biologist by craft. He's you are a full professor of microbial ecology at the University of California, Berkeley in the United States. So please also come up. Yeah, go. You're fine there. Then Dr. Ricarda Steinbrecher, the stone breaker, translated, is a molecular geneticist by profession, a developmental biologist based in the UK, working for her own research institute and consulting institute called EcoNexus as well. And another dear colleague of ours is Dr. Sarah Agapito, Brazilian by passport, moved to Norway now, working as a plant molecular biologist and a plant geneticist there, and studying the biosafety issues and the basics of how transgenesis works and how GM plants actually function since many years as well. So welcome as well to us, Sarah Agapito. And with that, I would like, who wants to go first to answer some of those questions of what you were thinking of it? And maybe do we need this, or do you remember the question, then we can turn this off, perhaps?
It would be nice to us, unless you...
Yeah, it would be nice to us, yeah.
Oh, then we keep it, okay, yeah. Then we keep it up there.
No, it would be nice to turn it off.
Turn it off,
okay.
He wants it.
Okay, we can see it here.
Okay.
Yeah, then
know,
just
should
the
we
light
sit?
goes off and you keep seeing this here. Okay.
Angelika, you're abandoning us?
I'm taking a back seat now because now you become the protagonist.
Can you move?
Yes, this way?
You can also, this way they can talk to each other.
Ah, yes.
So what is your thinking about these scientific foundations?
I think your talk was great. I think it was brilliant in the sense that it opened a whole range of new questions that, of course, we have been talking about for years. But I want to just honor Angelika's capacity to connect with you guys. Her capacity, what she always complains is these conversations that many people in biology are having today, because you could say that biology is in a crisis, in a conceptual crisis, that she did a really good job of presenting to you. And there are conversations going on, but her complaint is that you guys don't hear about it. So this is really the first lecture that I see where these questions are opened. They're opened in a beautiful way and in a way that I think is fruitful. Instead of dealing with questions about yes or no, pro and con, and so on, I think you raise the discussion to a very different level, and I welcome that. That's what I think.
That was a way to avoid the question.
Yeah.
Well, I am a geneticist, and I'm, all my professional life, a genetic engineer. So I very much appreciate what I see as both the power of genes and the power of the science of genetics. And all of my life, all of my professional life, I haven't been able to make a divorce between genetics and genes. So for me, it's an affront a little bit when genes become simplified as molecules, polymers of nucleotides. Now, I understand, of course, that there is a lot of evidence that certain traits require certain sequences of nucleotides in DNA molecules. I couldn't credibly say the otherwise as a geneticist. But there is no experiment that says all genes must be DNA. If you go back to the original experiments, Avery et al., who had shown that a molecule from dead pathogenic bacteria could transfer to live benign bacteria inside a mouse. And that material was susceptible to enzymes that destroy DNA. That is convincing evidence that the trait he was looking at, the responsibility for the trait he was looking at, devolves to a DNA molecule.
I would disagree.
It's okay. I won't leave you enough time.
Yeah.
And even he also didn't claim
Yeah.
that it was absolutely DNA, but I don't think that I need that right now.
You need
So, however, what his experiment didn't show is that all genes needed to be DNA. But very quickly, after Avery, was Hershey and Chase, who showed that it was the nucleic acid content of viruses that got into cells and reproduced viruses. This was all becoming a consistent line, that nucleic acids, particularly DNA, was the stuff of genes. And for those traits, it probably was. But none of these experiments could discover other kinds of genes. And when we quickly coalesced on DNA as an obvious molecule of genes, many of us stopped looking for other material bases of genes. We started sequencing DNA to find the genes. We didn't do experiments that found the source of genes. And that's where the field of epigenetics languished. But epigenetics actually predates Hershey and Chase and Watson and Crick. Thomas Hunt Morgan, the father of the chromosome theory, again, an intellectual precursor that narrowed the material basis to DNA, left that field in the 30s to return to his first love, epigenetics. So epigenetics has been with us a long time, but it has never been mainstream. And so for me, the issue as a geneticist is I want to understand the basis of traits. I don't necessarily want to restrict my search to DNA. And then we build regulations based just on a simplified view of genes. And then we have debates about whether only DNA matters when it comes to looking at the risks and promises of biotechnologies. So my view on this is that we've come too quickly and prematurely to a narrow view of what genetics is.
Yeah. That actually makes me sort of like think of a more general understanding of models, right? When we try to understand something, we try out different models. Like, would that work? Would that work? And experimenting with it, whether sort of it might explain what we are seeing. And they are helpful. Models are really useful. Depending on what you need them for, they might actually look different, even though they are about the same thing, right? But if you need a model for something that contains water, fine. But you might also want to have something that is a model for something that actually fractures light differently, right? So you can look at the same thing and develop different models with that. And models actually help us look at things, but they are not what they are, right? And that is important. I think that's where we got stuck, that we all of a sudden made the model of a gene, the model, like the dogma that was supposed to help us understand one aspect of it, that became the reality. The model we got stuck with. And it's very hard to then step out of it, because it's sort of like, where's the railing? What is reality if you all of a sudden, that what you grew up with doesn't exist? So I feel like that's, in particular, I think, like my generation will hold on very strongly to the idea of, what a gene is, what DNA is, and not want to let go, because that is what we were taught, and that also will do the direction of our research and of our predictions. And it's exactly where it shows that the model is wrong, because we can't actually do any predictions with it that actually come to life. And this is because organisms just don't work that way. They don't work in a way that you can just line up different DNA letters, nucleotides, and therefore you get a predictable trait, or in a way that you can just move it around sort of and think of it more like a machine, because it's a multi-interactive system. And actually it's incredible, sort of like thinking that it was described as noise. Sort of, God, they must have had really like multiple earplugs on, not to
No, I didn't.
But basically, if we don't understand how traits work, how, for example, good farming works, how do we get really a good crop? What do we need for that? Then we will actually get further and further away from our goals if we follow a model that actually is not the right model to understand traits. It's just a model to understand like a piece of string of DNA. Now, if we go and want to turn a plant into what is often being sort of suggested into a Olympic athlete in a way, so it can sort of get its own nitrogen, it doesn't need any water, although athletes mostly do, never mind. You get them like super fast, super yields, like how much you need to train for it, how susceptible really top athletes become to, some stomach upset, like they will not be able to produce properly then, or, they need a team of physiotherapists around. Well, that is what we will do if we are trying to find plants and put all the traits into the plant. It needs a whole huge support system to be its doctors and nurses, right? Whilst actually what we need is a resilient agricultural system that can actually support each other. And that's actually how it works, how we really get traits. And if you look at some scientific experiments that looked at higher yields, what you find is if you use a crop, and for example rice, and you plant next to it beans, right? And you count them, look at the yield, and you call that it equals 1, right? Then you plant them together in the same amount of field. You plant them together, then you harvest it and check what the yield is. And if it's higher than 1, then it means you have actually gone beyond your previous system. Well, what you can do actually, and there's lots of papers, whether you go to agricultural systems in Africa, in Latin America, in Europe, everywhere, in the US, there have been systems made with this land equivalent ratio, it's called, that compared it, and you go up to 1.7 just by mixing crop plants with each other rather than monoculture. Our research is going towards an agriculture with severe monoculture that is susceptible to diseases, but not that yields high on its own. So, but that is because one system is actually working with a reality and understanding how to get the best out of something, whilst the other one is trying to actually ignore how the system looks like and hangs on to a model. But
can I say something? So I think there's two main issues why they're bound to fail. So you have touched on the issues of productivity and the traits and what the promises are for the future of these crops. The other issue is their safety. And I see, for me as a molecular biologist, when I started this whole thing, I was trained in agronomy. I found it super interesting that you could actually make a pest-resistant maize that easily, That's what I thought in theory. But I also wanted to see how this transgene would regulate other genes within the maize. So one of the reasons why I think it's bound to fail is because there is a contradictory discourse in the industry regarding the technological advances we have. So one promise is that we have now CRISPR-Cas and all these new technologies that are safer, more precise, but they fail and they deny the use of these same technologies to test the safety of these crops.
Yes.
So it has been a fight among us that do the hardcore genetics to test for the safety and to include that in the risk assessment framework that are currently there. And I understand many claims of being very difficult, not mechanistic, like hard to implement, but that shouldn't be a reason for us to avoid to use what we have the most, the advances in technology to test for the safety. So that's another aspect of the issue.
I want to drag the conversation back to what Angelika is challenging us with. And I must share with you that in my teaching, I've been teaching at Berkeley for 22 years now. And in my teaching, I have figured a way to present biology without using the word gene.
Wow.
Yeah. Try it at
That's
home.
blasphemy.
Try it at home. It's really difficult. And it takes a lot of thinking around. Because what we were given, what I was given as a student, and what continues to be given to students in genetics classes, if you open any textbook in genetics for college, for university level, the first section will always be Central Dogma. Let's establish that, and what that means, as Angelika presented it, is you have a code, and it's DNA, by the way, have a code, and there is a system of translation into the real world of that code that gives you form and function, like Angelika showed, right? The shape of your face, the function of your body, form and function are really the same thing at different scales if you think about it. So there's this code that gets translated into form and function. Once you accept that, and once you believe that that's really the only way things happen, everything is great. Everything is so beautiful. You can build a castle that makes so much sense, that has all kinds of beautiful spaces and rooms of imagination of what you can do, what you cannot do. And I'm sure you've seen all the YouTube videos of molecular dynamics that they say this is exactly what it looks like, even though it's an animation in a computer, right? It's a model. But once you accept that, you are in a beautiful, beautiful, beautiful castle of imagination, which is the castle that is used to present stories of fear, be scared because this is so terrible, and hope. We are going to save the world with this. All within the construction of this thing that is founded on this one stone at the base of it. And it's that stone that I try to teach my students, my young students, to question. I don't say that's, I'm sorry, I won't use a bad word, that's bad. I don't say that is propaganda from the industry to brainwash you or anything like that, because it's not. It's really a very interesting model from the 1940s, 50s, and 60s, when we were growing up, that was very useful at that time to establish certain observations and use a model to try and explain how the world was. And as Angelika said, and Jack said too, the problem was that we never recognized, and you said it too, we never recognized that this was a model, that a model needs to be changed once you recognize the problems with it, and then you can move on to the next model. So this model got fixed. The most important part of it is that it got this word attached to it, that even as a student myself, back in the 70s, I was asking, why? Why do we call it a dogma? And there is a reason for it. Because you need to not doubt that, otherwise the whole castle comes crumbling down, which is what I think is really great about this conversation. Exactly. This is really what you guys, I think, are watching in real time. This effort to try and portray how this castle just breaks down. And I just want to say one more thing. I don't want to monopolize time, but I want to say one more thing for the moment, and that is, when you think about it, you don't need to know molecular biology. When you think about what I just said, you have code that gets translated through some kind of mechanism to give you the reality of the world. I think you would see it, you will see it very clearly as a very old, certainly medieval, but maybe older kind of idea. It's an idea that says there is a code that predetermines, prescribes in some kind of magic language over here in the sanctum sanctorum of the nucleus of the cell, and so on and so forth, prescribes what the world is going to be like. What an incredibly deterministic and old-fashioned medieval concept, right? That you will have a code. Somebody wrote actually the instructions for why it is that you are feeling the way you're feeling right now. Why you thought you would come here because you wanted, because you have free will and all these things. No, that was written somewhere in code, prescribed for you. So once you recognize that and realize that, at least myself, I realized the incredibly liberating power of rejecting that idea, of saying, I don't think so. And then when you start seeing what epigenetics, among so many other things, you hinted at the RNA world, and there's about 20 different lines of biological research that reinforce the idea that this code means reality in an encoded way really just doesn't work. When you recognize that and when you say, I'd rather go looking somewhere else than believing that somebody, some magic somebody, actually wrote the instructions for everything I am and everything I will do forever. So for me, this is an incredibly powerful way of looking at biology that is liberating from what has been, I believe, a cage in which we as a society, and certainly young scholars and new biologists coming into the field, have been caged for much too long. And I do not want to go to my grave without speaking out about it.
I'd like to.
Yeah, there was a question.
Yeah.
Okay, I can follow up with it later.
But she had a. No?
Well, there's two.
Let's finish your
Okay.
conversation, then keep the
Oh, we'll never finish the conversation.
That's the word.
Public soon.
Yeah.
Okay.
Let's be fast.
Okay.
No, exactly. So, which I was just reflecting on, sort of that in different times, in different cultures, we are strongly influenced by narratives, by what we think are the realities. Like they can also be ethical narratives. How one is supposed to behave. But like a narrative that we have running in the moment is that of technology as an answer system. Technology to help us fix problems. So it's technology fix. And it is, just check with yourself. You will have it also running in yourself. We have that quite deeply installed, and it's very difficult to step back from that. It really is difficult. We'll always think like, oh, how can I, which technology could I use to get that faster, or to deal with this? And so it's not a surprise that we think of technology also like in order to try to direct biological systems that actually functions different, but we try to put our ideas on it. And that doesn't mean that using genetic engineering, we cannot all of a sudden get a trait into it. Of course we do. I mean, simple things do work, you know. I mean, if you transfer a gene that actually is responsible for one trait, that's sort of like herbicide tolerance, and you put that into an organism, then spray, well, that gene is most likely, unless it was switched off, is most likely help that organism survive that spraying. So simple traits like that, you can do with that technology. But once you move into complex systems, it just will fail. And so my prediction would be that we will not really go far with this technology. There might be some more simple sort of changes or alterations, and why not? But it cannot be the answer, because the system actually, the model is not working in the complex system.
Can I just resurrect one point quickly? And it was really from Sarah, and I don't want it to get lost. And Sarah brought this around to what I think many of us are concerned with. Not what can be done with it, but how can it be shown to be safe? How can we assess these things to have some confidence in them? Should they deserve that kind of confidence? And Sarah made the point that we're not using the full tools available to us. That is, those who are developing these materials aren't using those full tools. But we're also sort of contributing to the problem by having all these words. So we have words like genes, and we have epigenes. We don't need epigenes. Epigenes now, I mean, when in Thomas Hunt Morgan's day, it was about how cells differentiated from a zygote into the beautiful things I see in front of me now. But it's become a word to mean not DNA inheritance. Well, we don't need a word for not DNA inheritance, because we already have a science of genetics, which is the science of inheritance. So let's get away with epigenetics. If we get away with epigenetics, then what we find is that there are going to be potentially lots of products coming through the pipeline that are using the escape route of calling these epigenetic changes to avoid regulation. And for us, or for me, that's, and inspired by Sarah, that's sort of the practical point of this. We create a diversity of language based on old and proper uses of words, or their narrowization with time and with the power structures of science. And then they get caught in regulation, and we lose sight of the point of the regulation. And the point of the regulation was that we are making changes to things that can reproduce independently of human intervention. And whether they can do that because it's based on DNA, or it's based on something that's not DNA, shouldn't matter for this conversation. So epigenetics and epigenetic changes, they're actually kind of a whole new frontier of possibilities for changes, but as well, they're a whole new opportunity for releasing changes that at least I would be more comfortable if we had evaluated as safe first.
Sarah,
Okay.
you had mentioned algorithms before. I think that would be brilliant if you put something in.
I think the question's first, no?
Yeah.
You could
If-
bring the algorithm in if you want.
If you want, have a last word before we go into the discussion.
Yes,
Go
well,
ahead.
yeah.
Yes?
So one of the issues that I am investigating right now is that much of these techniques, they are built on databases from genomic sequence, from model organisms. So you see the filtering.
From DNA sequences,
DNA
not even genomic.
Not even genomic. DNA sequence from model organisms. And this is our source of understanding the organism in front of us, which might not always be a model organism. Certainly not the organism that has been sequenced and deposited in a database. And what it seems to me is that whenever that relationship is established, it's really narrow. Because then what you see is just a reflection of what is in the database, because that's what you can track. And what you don't see, it's ignored. So, for instance, if you want to test for, if you are making a GMO, let's say a maize, that has a modification on its DNA content, and you want to check whether this new RNA content is going to have an impact on non-targeted species or the species that are there in the X system, you can only rely on the species that are in the database because you're not going to sample that. So that's uncertainty. And uncertainty has not been considered in its full concept within biosafety. So that's where the algorithms comes, because these databases, they are always also prediction tools. Like, okay, we don't have all the data, but we can predict. But the prediction is based on a ruling of the data that is there. So it's a loop that you never get away, you know?